Good morning, Mirabelle here from TELUS World of Science, Edmonton. And today I'm delighted to be joined with Dr. Houghton, who is the co-discovery of hepatitis C, currently working on a vaccine for that virus. And now with the advent of coronavirus, he is leading a team at the Shing Applied Virology Institute at the University of Alberta, looking to develop a vaccine against SARS-CoV-2. Thank you so much for joining me this morning.
It's my pleasure, Mirabelle.
So first question, let's take things back a step. Can you just quickly explain to us what are viruses and how do vaccines work?
Well, yes, what are viruses? I think viruses are genetic elements that have evolved and adapted to humans to replicate, reproduce. They use the human cell as their factory and reproduce themselves. And they've also adapted to stay in the human population.
Okay, so we've been hearing so much about the vaccine and how life will not be able to return to a so-called normal until we find a vaccine. Why is a vaccine so important?
So what you try and do with a vaccine is you basically fool the immune system into believing it's under attack by a COVID virus in this case, but the vaccine is not infectious. So as far as the immune system is concerned, it thinks it's seeing the virus, the infectious virus, but the whole point of a vaccine is to get the immune system to respond, but not with an infectious entity like the virus.
Okay, so that's very different than a drug or.
Yes, drugs. Excuse me. Drugs are there to treat patients.
Right.
And usually the drug is aimed at some component in the replication cycle of the virus. A drug will hit an essential protease of the virus, or it will hit an essential polymerase of the virus responsible for replicating. Mm-hmm. And so the drugs are used to treat patients, and we need those. There's a huge effort worldwide to try to find a drug that is already approved for human use for some other indication that might work against COVID. But the vaccine is there to prevent infection and disease in the first place.
So let's talk a little bit more about that. So your group is currently working on a vaccine. Can you take us a little bit through the process? How does one develop a vaccine? What needs to happen? What are the steps?
Yes. Well, traditionally, the way to make a viral vaccine is to grow the virus in large quantities in the lab, purify it, and then chemically inactivate it so that it's no longer infectious. And then traditionally, that's been the way to deal with viral vaccines. Now, to do that with COVID will take a long time because we've only just been able to grow the virus in cell culture. And then there's a tremendous amount of scale up and safety issues for the workers to produce an inactivated COVID vaccine. Fortunately, some methods have been developed in the last 20, 30 years, sometimes earlier than that, which are a bit faster and safer. So now it's possible to introduce part of the genome of the virus. So the virus, COVID, has a RNA genome of about 35,000 nucleotides. But you can take a small piece of that produces the virion protein, the spike protein it's called. It's that mushroom that is sprouting from the surface of the virion. So you can just take that piece, introduce it into the body in a certain way that it gets taken up by immune cells and expressed into that virion mushroom. So the immune system sees it and thinks, well, I'm going to respond to it because it's a foreign pathogen. Our bodies have evolved to do that in our immune systems, and they will start making antibodies to that spike, that mushroom protein. And those antibodies are capable of preventing infection of the virion. They bind to the virion, and then the virion can no longer infect cells of the body. So there are some clinical trials already in progress around the world using what's called RNA or DNA vaccines. And it's going to take a while to develop the technology to scale the virus production in cell culture to the extent where you have enough to give to people. But what we're working on, and what we've been working on in the Shing Institute of Virology at the University of Alberta for many years now, is a way to make a hepatitis C vaccine by actually making the proteins of the virus in the lab. So once again, we take a small piece of the viral genome. We just take the piece that encodes the surface proteins that the immune system sees, and we can persuade cell cultures to produce that protein. And then we scale up. We have technologies to scale it up and then purify it and then deliver that to people. So what we're making essentially is the surface protein of COVID, which is the spike protein, in cell cultures. We then purify it, and then we add a substance called an adjuvant, which revs up the immune system at the same time that you give the spike protein, and we stimulate the body to produce antibodies that prevent infection.
So one thing we've been hearing about a lot is the timescale needed for a vaccine. And obviously, you know, research takes time. You've also mentioned a little bit the question of scale. You know, if we need to immunize the whole global population, obviously, you know, manufacturing of the different components will take a lot of time. Can you give a bit more insight, though, as to what, so from what you're describing, it sounds like at the heart of a vaccine is basically replicating some part of the virus to trick the human body into believing it's been infected. What is taking the most time? Where do those timelines come from?
Yes, we're producing cell cultures right now to express the spike protein. Then, you know, that takes quite a while. You have to use a very clean cell line, and to select for a good expressor can take 2 to 3 months. So it's not something you can do overnight and the next morning you have it. You have to actually select for a high expressor that's stable. Then you have to purify the spike protein from those cell cultures. It's usually only 1% approximately of the total mass. So you have to purify it. You have to figure out a way to purify it. Then you have to get it clean enough for the regulators to say, okay, it's safe to put into humans. So Health Canada is the regulatory body in our country that decides if your drug or your vaccine is clean enough to put into humans, and it has to be very clean, of course. And so, without going into the details, we're going to have to prepare the vaccine in a very clean way. And then secondly, we're going to have to test it in animal models of COVID to make sure that it's protective, but also that it doesn't mediate any side effects. You know, that's why it's a somewhat frustrating situation where you want to protect people against this deadly disease, but if you go too fast and say your vaccine is contaminated with a bacteria and you didn't do the tests required to pick it up, you could end up, you know, hurting a lot of people more. Okay, it's preventing deaths against COVID, but you could cause a lot of damage in other ways. So it's a bit of a conundrum, I would say, a dilemma.
That's a really important message, I think, when it comes to vaccine, as you've so well highlighted. You want to make sure that the cure isn't worse than the virus you're treating. Because the stakes are so high, then obviously due diligence, all of the systems are in place. And from what I've read, a lot of things are being accelerated because of the gravity of the situation, but you can only cut so many corners until you get to a situation where you might be putting more people at risk.
Exactly. And Health Canada are very good, the regulating system in Canada. I should add that, the RNA and DNA vaccines that are already in clinical trials, it's very relatively easy to make those. Now, if they work, then we could have the vaccine earlier. Maybe the end of this year, beginning of next, we'll have enough vaccine to give to Albertans. But the question is, are they going to work as well as the protein vaccines that we're making? I hope they do, because that is the fastest way to deliver a vaccine to the community. But historically, those kinds of vaccines, they can work, but sometimes they don't work very well in humans. So we'll have to see. I do hope the RNA and DNA vaccines work, because that is the fastest route to protecting the community.
So talking about the efficacy of the vaccine, another big question that we've been hearing a lot about is just how long immunity lasts, which is a really important consideration when it comes to viruses. Yeah. Do we have indication so far as to someone who's either contracted the virus or someone who would get a vaccine? How long would they be immunized for?
Yeah. Well, great questions. Unfortunately, as you know, the outbreak in China only occurred in December, and we're in April. So
it feels like 10 years ago, but
it seems like a lifetime. Yeah, absolutely. But it's still early days, and we don't have all the information yet to be able to answer your question with confidence. But based on what we know about the SARS 2003 virus, which is about 80% similar to the COVID virus, so it's highly related, but it is distinct. But based on what we know about SARS and what we know about MERS, I think there's reason to be optimistic that we can make a vaccine to COVID, and that it will provide durable immunity. It's not like, as far as we know today, it might change in 2 months' time.
Or 2 days.
From what we know today, it's probably not like influenza, which mutates so quickly that each year we have to have a new vaccine. I cannot rule that out completely, but I would say right now it's likely that the vaccine that we and others are developing to COVID probably will protect most of the strains out there, and it's not something we'll have to be doing every year. And in terms of duration of protection, the whole point of a vaccine is, as I mentioned, is to persuade the immune system to make protective antibodies using a non-infectious vaccine. Once you've done that, our immune systems have evolved to have memory. So now the B cells that produce antibodies and the T cells that respond to infected cells, they have a memory component. So they don't just get induced and then disappear. They get induced, they start declining when the virus goes away, but some of them remain as memory cells. And when the virus comes back into the body, those memory cells are there to rapidly expand and protect us against reinfection. So the reason that doesn't happen with influenza is that every year there are new strains that are not protected by the previous vaccine.
Thank you so much for being with us today, Dr. Houghton. I really, really appreciate you taking the time and providing some some knowledge from the front lines of vaccine development. It's incredibly heartening to see all of the efforts that are happening both here in Alberta and the collaboration happening around the globe looking for this very important vaccine. Thank you
Thanks,
again.
Mirabelle. Thanks for your work. And also just want to end up by saying that I have a very talented team working on this in the laboratories now. Very bright young people that are really providing intellectual, and they're also providing the workforce under these trying circumstances. So it's nice to know in Canada we have people like this that can respond to epidemics like this.